All right. Thank you for the intro, Dr. Rooney. I appreciate it. Thank you, everybody, for being here today for my seminar. So as Dr. Rooney mentioned, I'm going to be telling you a little bit about part of my thesis research, which is titled Evaluating Introgression Sorghum Material while Exploring Genomic Selection as a Screening Method. So let's get into it. So just a little bit of an overview for my talk today. I'm going to give you an introduction into sorghum, as well as the sorghum conversion program. And then that's going to lead us into the new sorghum, what I'll call the sorghum integration program, the GUESS program. This kind of lays the framework for why we're doing this research and why it's important. And from there, I'll tell you about a little bit about our methods for what we were looking at and how we got to where we are now, from population development to genotyping, evaluating the hybrids that we've created, trying to do some genetic distance analysis, and then finally using genomic selection as a method of trying to screen some of this introgression material. And then we'll get into some of our results and conclusions. So like every good seminar, I got to give you a little bit of background on sorghum here, just so if you're not familiar. So sorghum's a grain crop grown in the US as a hybrid. It's originally from Africa, and where it's typically grown as a landrace or a hybrid. It's grown in a lot of different places throughout the world in different forms. In the US, it's primarily used as a feed grain. There is a small niche market starting to come up for food and other applications, but primarily a feed grain for the US. In 2020, there was about 5.8 million acres produced, and our top producing states are Kansas and Texas. So it's a diploid with 2N equal to 20. So a pretty good genetic system to work with. And hybrid production relies on a three-line cytoplasmic male sterile system, so an ABR system for producing hybrids. So I mentioned the sorghum conversion program, and I also mentioned that sorghum originated in Africa. So this tells you that most sorghum material is tropically adapted. So what this means is that most sorghum material is photoperiod sensitive, which in turn means that it's either not going to flower in more temperate environments, or if it does flower, it's going to be 8 to 10 foot tall. So it's not suitable for grain production. So what this then in turn resulted in is that when sorghum was becoming a crop in the US, there was very limited diversity. So it was speculated that sorghum could benefit from the introgression of wild ancestors, wild accessions that come from these photoperiod sources. So with that, the Texas A&M sorghum program started the sorghum conversion program in 1963. And the aim of that was to take tropical sorghum accessions and introduce dwarfing and photoperiod insensitivity to them through a back cross process, where the cross would be made in Puerto Rico, where tropical accessions will flower, and then they were selected on in Texas, where anything that was photoperiod sensitive would not flower. So this was the way they were able to then get the photoperiod sensitivity into these tropical lines to make them more usable for US sorghum production. So upon its completion in 2003, they had released over 700 lines, or converted over 700 lines in that program. And then following that in 2009, a reinstated sorghum conversion program was started. Excuse me. That just utilized markers to try to help speed up the process of recovering the tropical genome. So you'll see over to the right here, it's a very old picture, but it gives you kind of an idea what that looked like. On the right, you can see that photoperiod tropical accession. And then right here to the left of it, you can kind of make it out there. It's, like I said, it's an old picture, but you can see its converted version right next to it. So that's kind of just gives you an idea of what the conversion program was doing. But you'll see, so over 800 lines were converted and released by the SC and RSC program. And while you'd be hard pressed to find material that's been released that doesn't have SC material somewhere in its pedigree, if you start looking at what SC lines are showing up, you're only going to find about 30 of them that show up in those pedigrees over the last 50 years. So that kind of gives you an interesting insight into what's going on there. So while the importance is like undeniable, it's very clear that there's something strange, or there's something going on there that where some of this material is just maybe still too little bit wild, has too much of the tropical genome in it, the unadapted genome. So that leads into then this new introgression program known as the GUESS program, started by Dr. Rooney and Dr. Bob Klein. And so this leads to an alternative approach where instead of back crossing back to the tropical parent, we're going to back cross back to the elite adapted parent. So what this does is it decreases the contribution to the genome from the unadapted parent. And so this kind of helped maintain agronomic performance while still introducing some new genetic diversity, some new alleles into the lines that we're looking at. So this program was modeled on a similar program that was implemented in the Queensland Sorghum Improvement Group out of Australia. They're doing similar research to this. But you'll see there at the bottom, the importance of the next part of this is that 30 populations have been created, and that equates to about 3,000 some lines. So that's a lot of material to evaluate. So moving on, that leads us to the objectives then of this study. So we need to start evaluating some of this material and determine if within these introgression populations, do we have material that's applicable and relevant to a US breeding program? Is there things in here that are worth going after? And after that, then, as I said, we have 3,000 some lines. Logistically, we can't evaluate all that material in hybrid combinations anytime soon. It's going to take a long time to evaluate that much material. So we need to determine if there's a way that we can start pre-screening this material using genomic information in some way. So for this study, we used 2 BC1 NAM populations, and those are the male parents in the study. So you can see in the diagram on the right here how these populations were developed. There's 2 different exotic parents for each population. The exotic is crossed to RTX436, which is our elite recurrent parent. The F1 is back crossed one time to RTX436 again. And then from there, it's self to the BC1 F3. From there, those BC1 F3s were all either crossed to 03017 and ATX2928, which are both elite females in the program. And so these populations were generally selected at a population level. So with all the populations growing next to each other, these were the 2 populations that when my colleagues and Dr. Rooney went through, they said those 2 populations are strong from an agronomic standpoint as a whole. So they didn't go through and cherry pick out individuals. Everything in those populations was test crossed to be evaluated. Genotype data was, or GBS, genotyping by sequencing, was done on the inbred parents. After quality control, we had around 67,000 SNPs for use. Those were used to calculate genomic relationship matrices, GRM, following procedures described by Van Raden for the females and the males. And then you can then create the hybrid GRM in silico by taking the Kronecker product of those 2 matrices. And so this describes the process of creating an additive genomic relationship matrix. You can then make an adjustment for dominance effects following Vita-Seca, as you can see here. So that leaves you with 2 genomic relationship matrices for your hybrids based on additive effects and based on dominance effects. For evaluating the hybrids, they were evaluated in 2 environments. Well, actually 3. If you're familiar with 2021 College Station, you'll know that was not a good year. So we have two locations here for 2020 College Station and 2021 Bushland. They were grown as RCBD designs with 2 replications. You can see our checks here that were also present in the trial. So DK3707 is a commercial hybrid that's available from DeKalb. And then you can see the rest of these hybrids are just elite hybrids from within our program to give us an idea of how this material stacks up against elite hybrids in the program. And it should be noted that the 2 that I bolded here, those are the recurrent parent from the introgression populations crossed to the same females. So this will help give us an idea of, can these introgression males outperform their recurrent parent? And so the traits that we analyzed were grain yield, days to enthesis, and plant height. I used just a random coefficients model to check for significance of effects in the locations. And in order to get blues for further analysis, a mixed model was fit in which hybrid was a fixed effect and replicate is random. So in order to start trying to screen this material, as I said, with genomic information, we did a little bit of a genetic distance analysis. So first we plotted the additive genomic relationship matrix using principal coordinate analysis, just to give us an idea of what the population structure looks like, how those males relate to their recurrent parent and their introgression parent. And then genetic distance was calculated with tassel 5. And I used that data to then see if we could plot genetic distance versus yield and see if genetic distance from the recurrent parent or the introgression parent gave us any idea of what performance in a hybrid would look like. Following up with that, we decided we should try doing genomic selection with this material. So genotype effects are in the form of GBLUP kernels calculated with the GRM. For the single environment models, I used just M1, which is based on additive and dominance effects. Then we tried putting the blues together from the two environments to try to borrow information across locations, more or less. And when we did that, we used M1 based on just additive and dominance effects, and then also included a model that included G by E, that tries to model G by E to try to help borrow information across those locations. And you can see that G by E was modeled in the form of block diagonal matrices. So for the genomic selection, the models were fit using hierarchical Bayesian framework in DGLR and R. The cross-validation schemes we used were two different schemes that are kind of replicate what a breeding program could possibly be looking at to evaluate this material. It's kind of situations that you could possibly use. So one would be that you've just got new untested male lines. So we've got 50% of the male lines removed their hybrids. This would simulate that we made test crosses with 50% of the males from the population. We want to predict the other 50% and see what kind of accuracy we can do that with. CV3, the other cross-validation scheme, would be simulating that we've made all the test crosses with one of our populations. We want to know, can we use all of those test cross data plus part of one of the other populations, so just a small subset of another population, to predict the rest of that population that we only have a small subset of. So these are both pretty realistic ways of cutting down the workload of making test crosses to evaluate this material. So for grain yield, we use three different metrics of accuracy. The first being the normal one that most people would present would be the correlation between predicted and actual values. We want to see how does that actually relate then to the ability to select accurately in two different situations. So we also then calculated our accuracy in selecting the top 15%, it's basically picking the winners, and also our ability to drop the bottom 50%, so drop the losers. And then for plant height and days to enthesis, I just use correlation to assess accuracy there. Okay, so getting into some results. So just for our phenotypic analysis for our models that we fit here, you can see that in College Station and Bushland, hybrid is significant for all of our traits, which is good to see. And then our rep effect was not. We can see that our CV and repeatabilities are all within fine limits there. When we started looking at the prospect of combining data here to do a combined analysis, it became very clear that with only two environments that are very, very different from each other, it was probably not going to work very well to do a combined analysis. And you can see from the test down there below that Shapiro-Wilkes, they're fairly normal, but they still fail. And then homogeneity of variances with Bartlett's test also failed. So for that reason, we chose not to present a combined analysis here. I fit a combined model, but it becomes very clear that environment and G by E is just a very crushing factor when you have that much difference in environments. So just to visualize the blues here, you can see the distribution of yield from College Station and Bushland. But then something to note here is comparing the populations down below. So on the left, you have the two introgression populations. So these are the distribution of blues for one introgression population and the other. And then this is the distribution for our checks. And then on the right, you have the blue for DK3707, which is industry hybrid. And so it should be noted here that in College Station and in Bushland, there are individuals that outperform our checks. So this is what we're wanting to see here, right? But it's also interesting to note that when you change locations, it flips which population did or performed better, right? So there's clearly a very strong D by E effect going on there. But it's still interesting to note that we have individuals that can outperform our recurrent. So that's reassuring. That's what we want to be seeing here. But the problem that we have is that you can see there's a lot of material that does not. So that's the issue, right? There's a lot of material that does well, but there's also a lot of material that does not. So then we have plant. I just want to show you guys the distribution for plant height and days to flower here. This is more or less just to show you that these lines are agronomically relevant, right? So that for plant height and days to flower, you can see that the means for those are on par with our checks, or slightly earlier or slightly shorter. So this is just to show you that the material is relevant. It's not just outperforming, but then it's 10 feet taller, right? So it is applicable hybrids for a US program. So I mentioned wanting to look at the population structure here. And so what we can see when we look at this is that this is one of our introgression parents down here in the bottom left. This is the other one down here in the bottom right. And then our recurrent parent for these populations is RTX 436 up here in the center. And we can see that our populations cluster really well together, but they are shifted back up to RTX 436. And this is sort of what we would expect to see. But you can still see then within those clusters that there's quite a wide range of variability for how close or far they are from 436. But yet they were all selected for being agronomically relevant. But yet there's that wide distribution there. So this is what led to the next idea of using genetic distance from 436 or from the recurrent parent as a metric of assessing hybrid performance. And if you're looking at this figure saying there's a whole lot of nothing there, you're exactly right. It did not work. So this is kind of what we expected, to be honest. So I don't want to spend too much time here, but it's very clear that when you look at the distance of these lines, either from 436 or from their tropical parent, you can't really predict their yield. So moving on from there, we wanted to then fit genomic selection models and see if that was an effective method for screening this material. So just a reminder up there for what our two models look like. So we can see for grain yield here that with M1 additive and dominance effect, just in College Station and bushland. In bushland, we were able to explain more of the variation for yield with our genetic effects than we were in College Station. But with plant height, it was the opposite. So College Station, we were able to predict more of the variation, or partition more of the variation to our genetic effects than in bushland. And I didn't show days to enthesis, but the trends for plant height hold true for days to enthesis as well. So despite not being able to fit a combined linear model, as I mentioned before, I still wanted to see what would happen if we combine the blues from the two environments and try to allow the genomic prediction model to use some of that information across locations. And so that's what you can see here in the combined. And what becomes very apparent very quickly is that the environment effect is crushing in this data set. It just completely crushes your ability to partition variation to your genetic effects. And so this is not entirely unexpected, but that does kind of influence what we'd expect moving forward to our results. Okay, so when we look at some results here for the genomic selection, we can see in our single environments using an additive and dominance model, in College Station and bushland, our correlations came out to 0.17 and 0.485. So then what does this translate to? Again, this isn't completely unexpected, right? Because we were able to partition more variation to genetics in bushland. But what does that actually translate to then in your ability to select? So we see that to be able to pick the top 15%, we could do that with about 33% accuracy in bushland and about 13% accuracy in College Station. So we see that there's a pretty large jump in the ability to pick winners from that jump in correlation there. But when we move over to our ability to drop losers, we find an interesting result here in that we can drop losers in College Station with about 60% accuracy. But even when our correlation jumps significantly to bushland, we're still only predicting with about 70% accuracy. So you can look at this one of two ways. The way I look at it is, if you've got a good model, great, you can pick losers well. If you've got a bad model, you can still pick losers with fair accuracy there, or drop losers with fair accuracy. So that's an interesting finding moving forward. You can see over here for our correlations for plant height and days to flower. As I mentioned, we were able to partition more of the variation to genetics in College Station for those two traits, so not unexpected, our correlations there were higher. Not going to spend too much time there. So when we move down to our combined environments, as I mentioned, I did fit a model using just additive and dominance effects, and as expected, that didn't work, so I chose not to show it. But when we look at the model that incorporates G by E, we do see very small increase in correlations here. But what that translates to in reality is that it kind of slightly increased the ability to pick losers in the bad environment and to pick winners and drop losers. But when we look at what our better environment was from a single environment standpoint, it kind of brought our abilities down there. So it's sort of a half dozen one way, six another, to where it probably doesn't make much sense to combine environments here and try to borrow that information. But it was worth looking at. So this is our next CV scheme here, looking at CV3, which was using one population plus part of another to predict the rest of the other one. So what we see here is that in this first set is we're using population 22 to train in predicting population 48. So when we have 100% of population 22 and predicting all of population 48, we're still finding a correlation about 0.35. So this translates to being able to pick winners with about 19% accuracy and drop losers still with an accuracy of about 60%, which if you remember is pretty similar to what our other cross-validation was showing. Then what we find is when we include just 10% of population 48 to that then increases our correlation higher than what we were getting before. And it translates then to being able to pick winners with pretty high accuracy, but also to drop losers with much higher accuracy than what we were seeing before. So this is a very reassuring thing to see that this might be an efficient way to screen this material to where we can use just parts of one population with all of another to start making some predictions. But what we found is that it is population specific at this point. So we need to do some more looking to figure out what about these populations is making this happen, because when we look at the other way around where we use population 48 to predict population 22, we don't really see that big increase when we add part of population 22. So predictions are still respectable there, but there's something interesting about that other combination that increased the prediction accuracy that we got to look at a little bit further. So wrapping back around to our initial questions, we wanted to know, is there material in these introgression populations that's applicable for the breeding program? So we found that there's a lot of material that is not going to perform well for the breeding program, right? It's not going to be relevant. But we also found that there's a lot of individuals that do exist in those populations that is relevant. So the material is worth looking at further moving forward. So then we asked, can we pre-screen this material using genomic information? So we've revealed that genetic distance does not reveal any information. This was halfway expected. We found before that genetic distance can't predict heterosis. So it's not an unheard of result to see this. We also found that lower correlations equal a sharp decrease in your ability to pick winners, but you don't see that same decrease in the ability to drop losers. So even if you have a pretty poor model, you can still drop losers with a pretty fair level of accuracy. We showed that when our environments are very diverse, it's probably better just to keep them separate. These environments are so diverse anyway that we're probably going to be selecting material specifically for them. We're not going to be selecting material that's going to perform well in both. So we may as well separate them and predict them separately. So to conclude, genomic selection can be effective for screening these introgression populations moving forward based on these results. So with that, I got to acknowledge my graduate committee, Dr. Bill Rooney, Dr. Patricia Klein, and Dr. Robert Klein. And I got to thank the rest of the Texas A&M Sorghum group, the Sorghum Genetics group, and our undergrads. Thank you to the Sorghum Checkoff and USDA NEFA for all their support and funding. And with that, I'll take any questions.
All right, questions? You mentioned that you got like 60K SNPs, right? So out of the 60K SNPs that you're using, how many SNPs that actually have the wild introgression, and how many is just the alleles from their current parents or the known parents?
From the known parents. So I didn't look at that specifically. I guess if you go back and look at the kind of the population structure that we showed before, that kind of gives you probably a little bit of insight into how much one way or the other that they have. I mean, the closer they are to the tropical parent, I would assume that they're probably going to have more of the alleles from that parent closer back to 436, they're going to be more similar to 436.
Yeah,
So being that they're squunched back up closer to 436, it's like we said before, 436 is contributing much more to their genome than the tropical parent is. But that was sort of our goal.
So what I'm asking is that essentially you already know the interaction between your two known parents, right? The only unknown is where you have a SNP that includes a wild introgression. So is it possible to actually just use that information to construct a prediction model instead of including everything that you have already?
I don't have a good answer for you on that one, to be honest. I'd have to think about that a little bit further.
Okay.
If you want to talk more later, I'd be open to
Oh, yeah,
that. I don't have a good answer for you off the top of my head.
Other questions? Bob, nail them.
So you showed that you used two different training models, right? One where you assess half a population to guess the second half of it. And then there was the other method was training a whole population and then guessing another half of, right? So it sounds like there could be a lot of time difference and the input of time that you need to use those two different methods, right? And since time is so important, which do you think is a better training method, if in your opinion, just because it seems like time is so crucial as well?
Yeah, that's a good question. So in reality, the number of hybrids that you're talking about making, it's probably going to be about the same, I think, in either situation, the more I think about it. But the advantage is that we already have these populations that have been grown all together in a location. So now it kind of opens up a new idea of, can we start then just making small subsets of these other populations and using the data that we've already generated from these other populations to predict those, if that makes sense. And I think there's ways to do that, just speculating. So in my mind, the way I'm thinking about it, the one where we're using the whole population to predict parts of the other makes the most sense logistically. And I think that it'll work the best.
So you think it's worth the extra time to take the phenotype off of?
Well, that's the thing though. We've already got two of these whole populations that have been phenotyped based from this study. So there's some nuances there we have to work through, but I think it's some interesting opportunities there.
So Noah, the only question I have is, a lot of these you guys made crosses early generation, right? Do you think your prediction accuracy, whatever model you use, would go up if we had the ability you were testing with maybe something that was self-down an additional couple of generations? Are you seeing a lot of variability that's might be throwing off your yield estimates, et cetera? Or is the early generation the way to go, early generation hybrid testing?
In my mind, I think the early generation testing is okay. I mean, being that these are BC1 F3s, they're effectively an F4. Typically, we start evaluating material in test crosses around the F5, and we don't see too many issues there of weird things happening. And just from looking at the material, I can tell you that for the most part, most of those hybrids are pretty stable. There's not a whole lot of instances out in the field where it's clear that there's something segregated in there. So,
All right. Let's thank our speakers again.